everyone, welcome back to my channel, and today we are going to learn pH in our topic enzymes. We are going to focus on the pH, and we have discussed other factors in the other video. Let's start with pH. So what is pH? Keep in mind H is always capital, P is always small. What is this? This is actually Puson's dehydrogen, which is actually the concentration of hydrogen ions that actually determines the acidity or alkalinity of a substance. So pH actually measures how acidic a substance is basically. When we talk about pH, so we have discussed this earlier as well, that there are different environments in our own body. Our mouth has a neutral pH that is around 7. Then our stomach has an acid, so it has an acidic pH which is 2. And then when we move to our intestines, the pH is 9. So we have different pH in our body. So how how one enzyme like is going to act at a certain pH. Now, whatever enzyme we have in our mouth, so that is amylase. Amylase is going to act in a different way. It acts on starch and converts starch into maltose or the smaller units. Okay, when this enzyme move into our stomach, it's no longer functional. Why? Because the pH of our stomach is now acidic. The enzymes that are actually functional in our mouth are not functional in our stomach. Similarly, if we talk about stomach, the pepsin is produced in our stomach in an acidic condition, and it's not going to work in intestine because intestine has an alkaline pH. So when the pH changes, you can say actually the structure differs a bit. You must have known that if we talk about amino acids, because these are proteins, amino acids have two ends. One is the like NH2N, that is a amine group, and on the other side it has the carboxylic end. So this is the reason that why they act on like different in different pH. So if you say there are some amino acids which have double amine group, there are some amino acids which have double carboxylic acid group. So that's why the pH is different for like different regions, or the enzymes are very specific to pH. So each enzyme has an optimum pH. Like we have talked about the amylase that is in our mouth, and then we talked about pepsin that is in our stomach. If there is an extreme condition, if you just change the pH of these systems, definitely the enzyme is not going to work. The structure of enzyme is lost, the tertiary structure is lost. There would be effect on hydrogen bonding, and if there is an effect on hydrogen bonding, then it leads to denaturation. So basically, the hydrogen ions and hydroxyl ions break hydrogen and ionic bonds in enzymes tertiary structure. This line is very important. This sentence is very important, and you need to learn that, and you need to keep in mind while writing the answer for how pH affects the enzyme activity. So these two bonds are very important to remember when we talk about the denaturation, because they are going to disturb if the acidic condition or the alkaline condition, that is, you can say the concentration of hydrogen ion or the concentration of hydroxyline is going to change. So if the shape of active site changes, there would be fewer enzyme substrate complexes, and we say the overall reaction is really low. If the structure is completely destroyed, or like the bonding that actually holds the tertiary structure, that is hydrogen bond and ionic bond, if that has completely vanished, if those are completely vanished, then there would be no reaction. Simple. So let's go to the graph. So we have, for example, two ones. One is the peptidase and other one is maltase. Now this one, let's say this act in an acidic condition. For example, in your stomach, where the pH is 2, and this maltase act in slightly alkaline condition. So we say the pH is, let's say, 8 over here. So when these extremes are changes, when these extremes got changed, then the reaction is not going to happen. Let's say this one is your peptidase, enzyme number one, represented with green line. So the activity will increase when the pH is increased. Let's say from 0, it's increasing to 2. So at 2, the you can say the rate of activity, or the rate of reaction, or the enzyme activity is maximum. But till 5, it actually denatures. But if we talk about maltase that needs to be active at 8, it's the activity increases when the pH increases, and it is optimum at 8 pH. But beyond this pH, the activity is going to decrease because your enzyme structure is lost. There would be no hydrogen bonding or any bond that hold the tertiary structure. And if the tertiary structure is disturbed, definitely there is no active site available. So now let's discuss about what we need to keep in mind when we investigate the effect of pH uh, or like effect of pH on enzyme activity. So we have to maintain a buffer solution if we want to check, because in the previous slide we have discussed that every enzyme has a specific pH, and that pH needs to be you can say maintained. So if this pH, like we say, it's a buffer solution. What is buffer solution? The buffer solution is that one which maintains its pH if there is an addition of acid or an alkaline, but in a small amount. We are not saying we are adding in a very high amount. So if there is a slight change in the external environment, this buffer solution keeps the you can say keeps the pH constant, so that your reaction is not going to disturb. So we have to have a buffer solution to keep the pH constant, and we use the buffer solutions of various values. For example, we have a buffer solution for 2, we would have the buffer solution of 8, and the same volume of buffer would be used in each reaction mixture. Simple. Now measure the reaction rate at each pH, and you will check whether it is going to change or not, and whether the rate is high, rate is low, or whether there is no change in the concentration of substrate or product. So a graph is represented between or drawn between enzyme activity and pH, and you can easily check whether it's increasing or is dropping. Simple. So we'll discuss the substrate and enzyme concentration in the next video. Stay tuned. See you in the next video.